talk a little bit about mutations. Now, we talked a little bit about this in some of our previous units. However, we're going to take a look at this specifically because now we understand a little bit more about the translation process, transcription process, and see where some of these mutations can occur. So remember way back in DNA replication, we know we have a possibility for DNA to possibly mutate in that particular location, so in the S phase of the cell cycle. But during translation as well, we could have some sort of DNA mistakes or possibly some other ways, such as mutagens, that could affect some of that base pairing. So, a mutation, as we've talked about before, is a change in nucleotide sequence. So, instead of the A, C, G, blah, 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 sometimes maybe one of those base pairs will actually change, or maybe a base pair won't pair correctly with its supposed complementary base pair. So, a mutation is just a change in nucleotide sequence. This usually results in incorrect proteins that do not perform the desired function. And as we've seen in talking about transcription and translation, those codons and those anticodons are extremely important to have that correct 3 sequence for codons in order for the correct amino acid to be placed where it should be. So the causes of mutations can vary. Sometimes we have mistakes in base pairing during DNA replication or RNA transcription. So remember, sometimes that could occur. But sometimes we have things such as mutagens. These can be things out in the environment, such as radiation, like UV rays or X rays. Sometimes X ray technicians, people who work out in the sun, a lot of times they could possibly be exposed to radiation that typically people that don't have those types of jobs can get sometimes as well. Sometimes chemicals can be mutagens, high temperatures. So people who have certain jobs or might be exposed to some of these mutagens could have a possible mutation occurring in their DNA. So let's talk about some of those specific types of mutations. Again, we've seen some of these, but now we are able to talk about them in a little bit more depth. So our first one that we're going to talk about is a point mutation. So just as the name suggests here, point mutation, this is just a simple base substitution. So this is a mistake in a single base pair. So this may affect the resulting protein just by changing a single amino acid. And sometimes that could have devastating effects. Sometimes it's just even though it just changes one letter, sometimes it is still the same amino acid. So an example could be that an A might be inserted instead of a G. Now, this could have effects, but sometimes it doesn't. So let's take a look at this example here, and I'll change to the pen so we can see. So a normal mRNA sequence might read this with a couple G's, C's, blah, blah, blah. But let's say that particular G here, so we have this G and this normal mRNA, that has now been replaced by an A. Now, if we were to read our codon chart, we would see that the protein or the amino acid should have been GLY, glycine. However, because we have that A inserted instead of G, we actually will get serine. Now, serine is a different amino acid. This could change the entire peptide chain or the protein chain, and it could cause some devastating effects. It might not, but it could. So, one single base pair mistake could have an effect on what type of amino acid is placed in the sequence. A second type of mutation is something called a frameshift mutation. So again, a frameshift mutation sounds just like the terminology, frameshift. So this is an addition or a deletion of a single nucleotide. Now this is actually going to shift all the other base pairs or all the other bases either up or down. So This affects the resulting protein by changing the entire amino acid sequence after the addition or deletion. So let's say, for example, here we have an extra U. So a U, a uracil, has been added in transcription. It just sort of implants itself. So we have this RNA sequence here, AAG, UUU, GGC. Now we've added a mutation, which is this extra U. So instead of having 3 U's, now we have 4. So we've added an extra U. So with our base pairs, or I'm sorry, with our codons, remember we have 3 of our base pairs will now code and be our codons here. And now what we have here is lysine, phenylalanine, and tryptamine. Now, previously our codons looked a little bit like this, and so we have the same 3 U's. So the first 3 codons are exactly the same, and they will code for the specific amino acids that we see here. We have lysine, phenylalanine. However, because we've inserted this U here, instead of the GGC codon, we now read UGG, which is a completely different amino acid. So it has actually changed the amino acid. So we're shifting that whole sequence down one, and that is affecting the amino acid that we have here. An example of a frameshift mutation is sickle cell anemia. So in here, we have what should be the sequence of CTT. Now, CTT on our DNA strand will read on our mRNA strand GAA. So this is our codon right here. So the codon, and remember with our talking about translation, we have our tRNA come in. The normal hemoglobin amino acid should be this, GLU, glucine. So in fact, what happens is with sickle cell anemia, we have this middle base has changed. So this middle base has changed from what it should be, read T, and now we have A. So what happens when transcription occurs, we have GUA instead, which is going to code for a completely different amino acid. And so this is the reason for sickle cell anemia.